In this video, we're going to explore the remaining assumptions that Mendel made when he was doing his experiments with the pea plants. And again, these are assumptions that simplify genetics, but in reality, there are many exceptions to these assumptions. So we'll take a look at what these remaining assumptions are and what the exceptions to these assumptions are as well. Again, always review your important terms before you come to this video. You should know what a trait is, you should know what a character is, and that many characters are heritable, meaning they can be passed down from generation to generation. So in the other videos that you would have watched, you would have seen that there are many assumptions that simplify genetics, and you can see these assumptions that are listed over here. Some of the ones that you would have seen already are that one assumption is that alleles show complete dominance, but we know that's not always the case. Sometimes we see incomplete dominance and codominance. Another assumption is that alleles only have genes only have two alleles. There's only two different versions of genes available for every character, but we saw that's not always the case either. For example, with blood types in humans, there are multiple alleles that control the blood type phenotype. So those are two that we focused on. Another one that we focused on was that genes are carried on autosomes, but we saw that that's not the case. They're not always carried on autosomes. For example, you could have genes carried on sex chromosomes, like the X chromosome or the Y chromosome. Another assumption, which we'll also see in this video, but you've already actually seen, is that gametes are made by normal meiosis, but they're not always made by normal meiosis. Sometimes there's errors that occur, and that could lead to damage to chromosomes. It could lead to damage to more chromosomes or fewer chromosomes in the gametes, and as a result, more chromosomes or fewer chromosomes in the organism that's made, in the zygote that's made. So whenever those exceptions occur, we might not see the ratios that Mendel saw when he was doing his experiments with the pea plants. We might not see the 3:1 or the 9:3:3:1 phenotypic ratio, or the we might see something more modified. We might see, for example, a 1:1 phenotypic ratio instead of 3:1. So those exceptions that occur would skew our Mendelian ratios. Mendel was fortunate that he didn't have those exceptions occur, and that the majority of his experiments ended up showing him a simplified view of genetics. Still very important to allow us to understand the concepts. Once you understand the basics, then you can see the exceptions and the complexities in a bit more detail. So right now we're going to take a look at this other exception here, which is this other assumption, which is that genes, or that characteristics, are coded for by only one gene. So for example, let's say we're looking at flower colors, so red flowers versus white flowers. What we're assuming here is that there's only one gene that controls flower color, and that one gene, might have two alleles. There might be a red allele version of that gene and a white allele version of that gene. But is it really always the case that characteristics are only coded for by one particular gene, by one particular locus? So what we're going to do is we're going to take a look to see if there are any exceptions to that rule. As it turns out, what Mendel studied, where you have one gene that controls one characteristic, that's called monogenic inheritance, where it's one gene that controls one characteristic. But that's not always the case. There are actually many characteristics that are controlled by many genes, not just one gene. So many genes on maybe the same chromosome, or even on different chromosomes. Many genes on different chromosomes might control for a particular characteristic, for a particular character. Let's just call that C. A few examples of that would be the height in humans, or skin color in humans, or even eye color in humans. That's not simply governed by a big R, little r. There's actually many genes that interact together and help to contribute to the appearance or the phenotypic expression of that characteristic. Usually when you have traits that are governed by several genes, we call that polygenic inheritance. Polygenic means many genes influence that character. So usually when you have polygenic inheritance, what happens is you have continuous or quantitative traits. In monogenic inheritance, you have what's called discontinuous or dichotomous traits. That basically means either or. So you're either red or white, or you either have widow's peak or you don't have widow's peak. There's no in between. There's no gradual in betweens of widow's peak to non-widow's peak. That's called discontinuous traits, and it's governed by monogenic inheritance. In the cases where you have polygenic inheritance, many genes that affect the characteristic, you end up with what's called continuous or quantitative traits. Continuous or quantitative traits, where you have many different variations of that characteristic. So for example, for skin tone, you can see you have very, very light, and as I go up here, you can see I get a darker and darker and darker and darker and darker, and eventually very, very, very dark on the other end. So I go from light all the way to dark, but I'm not white or dark. There's a range of variation instead of just one or the other. Same thing for height. You're not just short or tall. There's variations in height, and there's a range of variation. Some people are 5'7", some people are 5'6", some people are 5'5", some people are 5'5¾", some people are 5'8", some people are 6 feet, 6'1", 6'2", 6'2½". So when you have continuous variation, when you see there's a range of variability in that characteristic, you can probably guess that your characteristic is going to be governed or controlled by polygenic inheritance. Many genes are involved in interacting together to give you the height or the skin color or whatever it is. It's the same idea for when you see an either or trait, you know that it's only one gene. So here we have the example of skin pigmentation or skin color in humans. That's actually controlled by 60 genes. So there's 60 genes that control the skin color. And those 60 genes, because there's so many genes that are involved there, give you a variation, a wide range of variation with skin color. It's not just white or dark, it's this whole range. All these different colors show the different ranges of skin color that you can have and skin pigmentation that you can have. And so I'm going to show you a simplified model here. The idea of skin pigmentation is that there's two different alleles that you can get for every gene that controls for skin pigmentation. So we're going to assume that there's only 3 genes that control skin color, and that's just very simplified because there's actually 60. But let's just say that for every dominant allele that you get, big A versus little a, you get 1 dosage of darkness, and for every recessive allele you get, well, you get a dosage of not darkness. You don't get any pigmentation added to you. So let's say you had this individual with all the dominant alleles, this would be the darkest you can get, and this individual with all recessive alleles, this would be the lightest you could get. If they had kids, they would have all heterozygous individuals, and those heterozygous would have 1, 2, 3 dominant alleles, and so they'd be kind of somewhere in between the white and the darkest, the lightest and the darkest. And then if you had 2 heterozygous individuals have kids, well, there'd be different doses of skin pigmentation given to them. You'd have some kids that get no dominant alleles, and so they would be the lightest. Some kids that get all dominant alleles, and so they would be the darkest. But then you'd have kind of some in between. You'd have some that get maybe 3 dominant alleles, or 2 dominant alleles, and so these 2 are going to be dominant then, more dark than the ones that didn't get any dominant alleles at all. So every dominant allele that you get for each gene that contributes to skin pigmentation gives you a dosage of darkness. If you have more dominant alleles, you'll be darker than if you had fewer dominant alleles. So for example, this one here that has 6 dominant alleles is going to be darker than someone that only has 3 dominant alleles for skin pigmentation. But the idea is that it's the interaction of all these genes, gene A, gene B, gene C, that determine your skin pigmentation. And that's why you see so much variation in the skin color characteristic. It's not you're either dark or you're light. It's a continuous variation. If you were to plot all this on a graph to see what proportion of the population is dark and what proportion of the population is white, you would actually see quite an interesting trend. If you look over here, you can see the characteristic curve for continuous variation. This is called a normal distribution. And what you would see is you would have, at the most extreme ends of the phenotype, the fewest number of individuals. So the lightest, you'd have the smallest number, and the darkest, you'd have the smallest number. But then as you go to the intermediate phenotypes, you'd have a larger number of individuals that have those intermediate phenotypes. You're not as likely to get only recessive, and you're not as likely to get only dominant. You're more likely to get somewhere in between the two when those genes are passed on. And that's the type of variation that you'll usually see in the population for polygenic inheritance, which results in continuous traits. So again, you would see this on a normal distribution. You might also hear it called a bell curve. Now, as you're going through these slides, it might be important to do some of the practice questions that you see listed. So for example, which person will be darker, this person or this person? There's a few ways you can do it. You can count the dominant alleles. So this person has 1, 2, 3 dominant alleles, and so does this person. So assuming that all the genes equally participate in the determination of skin color, then you would probably say that these would have an equal skin pigmentation. So we're looking at that heterozygous individual here. So let's find that heterozygous individual. So you might see someone with 3 dark ones, and then you might have someone, looks like this person here would be this person over here now that I'm looking for, would probably be somebody with 3 as well, but not 3 of each. It looks like it would have 2 dark and then 2 dark on the A gene. So let's say, for example, this is my heterozygous. I'd have the A gene, B gene, and C gene. I'd have someone that's more like this here. I could say this is the A gene, B gene, and C gene, and they'd have roughly the same skin pigmentation because they all have 3 dominant alleles. Now again, this is assuming that each gene contributes equally to that skin pigmentation. That even adds another layer of complexity. What if some genes contribute more than other genes? And again, like I said, if you were to plot skin pigmentation on a curve, you would see there's a great range of variation for the skin pigmentation color, and the curve that's formed would be a bell curve or a normal distribution, with the extremes being at the lowest end of that frequency and then the intermediates being higher than the extremes. So what we saw there was the assumption that we used to make, that characteristics are coded by only one gene, but as we know, characteristics like height, skin color, and all that can be controlled by more than one gene. And if that's the case, we don't just get either or traits, we get at that normal distribution, we get a variation, a good range of variation in that characteristic. Another assumption that's often made is that genes control everything, and that's absolute, that it's only the genes that determine what you will look like. In other words, they're not affected by the environment. So one assumption that many individuals make is that genes are not affected by their environment, they're unaffected, which we may see is not the case. We'll take a look at a few examples where genes can affect your characteristics, where, the environment can affect the gene expression. So here we have 2 plants, and they're actually genetically identical to each other. They have the same genotype in terms of the flower color, but you'll notice that they have different flower colors. And so the reason they have different flower colors, even though they have the same genotype, is because of the environment that they were put in. This one here was put into a basic environment in terms of acidity, and this one was put into an acidic environment. And whether you're placed in a basic or acidic environment for some flowers will determine what color their flowers are. So these are the hydrangea flowers, and their flower color is greatly influenced by the environment despite their genotype. So genes do affect what you look like and what your phenotype is, but they can certainly be affected by their environment as well. Some genes might be affected more by their environment than others, but the point is that that can happen sometimes. So if Mendel had been dealing with flowers that had genes which were affected by their environment to a greater degree, he might have been a bit more confused, or may not have seen those patterns as clearly as he did with the pea plants that he chose to study with. I'm sure you can think of other examples of genes that can be affected by their environment. I mean, if you think about it, height is a great example. If you have genes to be tall, well, how tall will you actually grow if you're not given the proper nutrition and vitamins and minerals and care when you're a child to reach that maximum height? So you need to think about the idea that yes, genes are important, but so is the environment, so is your health, so is what you put into your body in order to allow you to reach that maximum potential. So here we can see another example of an environmental influence. Siamese cats. So here we have a picture of a Siamese cat over here. And you probably noticed that there's interesting color distribution on the Siamese cat. You can see these dark regions on the face, the ears, and a lot of places on the extremities. So these paws over here and the tail. Yet in regions like the core here, it's very, very white. And so the reason for that is that the body core is usually hotter and the extremities are colder. And so the enzyme that controls for the color of the pigment on this cat here are actually sensitive to the temperature. So while all the cells in this cat have the same genes, you'd think that they should be the same color, but they're not. It's really because of that temperature sensitivity of the enzymes. In the warmer regions of the cat, the enzyme doesn't work as well. So there won't be as much skin pigmentation added. So it's made, but it just doesn't work well. And so that's why you don't have a darker area there. Whereas at the extremities, it's colder, the enzyme's made and can work well and therefore can add pigmentation to that. Here we can see another example in humans, cholesterol level. Cholesterol level oftentimes is genetic. Some people are just genetically predisposed or genetically more likely to have high levels of cholesterol in them and buildup. But it can also be influenced by your diet. You might genetically be a very, very healthy person, but if you have a very, very poor diet, well, you might have that cholesterol build up in you and as a result suffer from things like a heart attack. Or you might be someone who's genetically predisposed to maybe having cholesterol build up, but because you eat in such a healthy way and you exercise so much and you watch what you put into your body, you don't have that cholesterol buildup. So just because you're given a genetic hand, it doesn't necessarily mean that it has to determine everything about you. I mean, it does, but you can have a say into it by controlling that environment as well. So now we've seen cases A, B, C, D, E, and G as well. We're going to take a look at another very important assumption that's actually come to light quite a bit in these days, and that's the idea that your genes are carried on nuclear chromosomes. When I say nuclear chromosomes, what I mean is this. Here you have your cell, you have your nucleus, and whenever you ask any grade 10 or 11 student where the DNA is, they always say in the nucleus. And yeah, that's where your chromosomes are. That's where most of your genes are, but there are other genes that exist in your body, and they are not always in the nucleus. They might be in other parts of the cell. So one assumption is that all your genes are found on nuclear chromosomes, but today we know that that's not necessarily the case. There are genes found on other chromosomes that are found not in the nucleus, but other organelles or sometimes even in the cytoplasm of your cells. And so we'll take a look at some examples of that. So as I mentioned on the previous slide, most genes are found in the nucleus, but there are some genes found in the cytoplasm, and those are called cytoplasmic genes. And there are also some genes found in other organelles in the cells. So if you're dealing with, let's say, just humans or animals, you will find that there's DNA in the mitochondria of your cells. If you're dealing with plants who also have chloroplasts, so plants have both mitochondria and chloroplasts, they also have DNA in their chloroplasts and other similar organelles that we group together as plastids. But the idea is that genes are not only found on the chromosomes in your nucleus, they can also be found on chromosomes and DNA of organelles like your mitochondria or in the cytoplasm. Now, the DNA found in these organelles, they're different from the DNA found in your nucleus. The genes are carried on structures called plasmids. So in your nucleus, the DNA is more linear, like a string that's not tied at the end, so a linear chromosome. But in your mitochondria, you'll find them on these circular chromosomes that we call plasmids. And that's how we find DNA in bacteria in the form of plasmids as well. But I want you to get that there are still genes on these circular chromosomes that we call plasmids, and they can be passed on from parent to offspring in a similar way to how they can be passed on like the chromosomes in nucleus, with some exceptions, with some differences, of course. Another interesting thing about these plasmids, this DNA, this circular DNA, is that the organelles that contain these plasmids, they actually reproduce their own genes. They reproduce their own genes and they can pass them on to daughter organelles. So your mitochondrion might make 2 more mitochondria, and then those mitochondria would have the same plasmid that this mitochondrion had over here. So it can actually go and replicate its own DNA in an independent way from the nucleus, which is quite interesting. So just, you might see this in your textbook. Genes that are not found in a nucleus, that are outside the nucleus, are called extranuclear, extra meaning outside of the nucleus. So extranuclear genes outside the nucleus. The big idea here to get is that, yes, most of your genes that control for your characteristics are found in the nucleus. They are nuclear chromosomes, but there are some that are extranuclear. Some are outside the nucleus. Some could be in the cytoplasm, called cytoplasmic genes. Some could be in mitochondria, if you're a plant, in mitochondria and chloroplasts and other plastids. And when the DNA is found in the cytoplasm or mitochondria or chloroplasts, we find them on circular chromosomes called plasmids. They're not going to be on these linear chromosomes that you're used to seeing in your nucleus. And the organelles that have their DNA, they can replicate their DNA independently of the nuclear chromosome. So they can do their own replication at other times. So again, notice that difference here. We have circular for mitochondrial DNA, the mtDNA. So even though I didn't tell you what it means, hopefully you can kind of deduce what it means, versus nDNA. I've been saying these words. So mitochondrial DNA and nuclear DNA. I want you to start getting that into that mindset of, I don't have to tell you everything for you to deduce or to come up with what it means. mtDNA, what could that mean in this context? Oh, mitochondrial DNA, nuclear DNA, nDNA most likely. So nDNA, nuclear DNA, is linear. MT, mitochondrial DNA, is circular. So there's a difference in terms of the shape as well. But the idea for the expression is similar. If there's a gene on here, it has nucleotides, there's a code, a sequence, it's read by ribosomes, it makes proteins, and it gives some kind of characteristic. Now where it gets interesting is inheritance of these extranuclear genes. So the chromosomes that are in the nucleus, they're passed on by both your mom and your dad. Your mom gives you half of your chromosomes from the nucleus, and your dad gives you half the chromosomes from the nucleus. And that's essentially the general idea behind it. But when it comes to extranuclear genes seem for the most part to follow what's called a maternal inheritance, maternal, mother inheritance. So what that means is, whether you're a boy or whether you're a girl, most of your DNA in mitochondria and, let's say, cytoplasm comes from the mother. So you get most of your DNA from that's inside your mitochondria and your cytoplasm from your mother. Because remember, the egg is a lot bigger than that sperm, and it has a lot more cytoplasm and organelles in it than the sperm would. So the idea is that if there's any genes in mitochondria that are passed on by your mother, you're going to get those genes. But you won't get them from your father. So I'll show you an example here. Here we have two, these are called pedigrees. It just shows you characteristics passed on by mother and father. Here we have a mother affected by a disease, and that disease happens to be in mitochondria, her mitochondria. When the mother is affected by the disease, you'll notice that the kids are also affected by the disease. How do I know that? Look at the legend. This means affected here. Now, let's take a look at the father. If the father is affected by the disease, but the mother's not affected, notice the kids don't get the disease. So again, we're assuming this is a mitochondrial disease. Why don't the kids get the disease? Well, because the father's not passing on his mitochondria DNA to the kids. It's the mother that's passing it on. So in the case of mitochondrial DNA and genes, mother passes it on to the kids, not the father, because that egg has a lot of cytoplasm, a lot of organelles in it. So here's a few examples of conditions you might see associated with mitochondria. We have mitochondrial myopathy, which causes a lot of weakness and muscle deterioration, and if the mother has that in her mitochondria, that can be passed on to the kids. We have Leber's hereditary optic neuropathy, which is kind of scary because it leads to sudden blindness in people between the ages of 20 to 30. And again, if the mother has that in her mitochondria, that can be passed on to the kids. If the father has it, then it won't be passed on. There's a lot of research still being done on this. We think that these mitochondrial mutations can be involved in diseases like Alzheimer's, some heart diseases as well, and maybe even some cases of diabetes. So the more we understand this maternal inheritance of mitochondrial DNA, the more we can start to understand these conditions and maybe interfere and hopefully find cures and preventions for these diseases. Now, what's interesting is, again, this is the majority of mitochondrial DNA is passed on to the kids from the mother. There are some rare cases where, and this has come to light recently, that maybe the fathers can pass on some mitochondrial DNA. But for the most part, it's going to be, at least right now, what the research is telling us is that it's maternal inheritance primarily. So mostly the mother passing on these genes. In rare cases, father. I'm just letting you know that because that's recent research. But again, it's primarily the mother who passes on these mitochondrial genes. Here you can see an example of a condition caused by DNA found in chloroplast, and that would be from the female. Female gamete that passes that on. If the father had the DNA that causes this, let's say the sperm or the male gamete, then the plant would not have this variegated color on its leaves. So we've seen A, B, C, D, E. We've seen H as well. So these again are the assumptions, and we've seen the exceptions. We've seen examples of exceptions to each of these, which would lead us to a ratio that's not quite that 3:1 or 9:3:1 ratio that we're normally used to seeing. Another assumption that we've also seen, but I've included it in this PowerPoint because it is part of the assumptions, is that gametes are made by normal meiosis, but we know that's not the case. We've seen non-disjunction, so I'm not really going to talk about them too much. I'm just going to go through the slides quickly to make sure that you review them, but I won't talk about them too much in this presentation. You can go back and see the previous presentation on non-disjunction. So again, mistakes can happen in meiosis, and that could lead to differences in chromosome numbers, but also chromosome structure. Chromosome number might be you have missing chromosomes or extra chromosomes, and chromosome structure is you have the correct amount, but you might be missing a piece of a chromosome, or you have additional pieces, or the genes are inverted, and you've seen those already. Remember, non-disjunction is when an error occurs in meiosis, and that non-disjunction can happen in meiosis 1 or meiosis 2 and lead to different results. But what I want you to remember is that it could lead to abnormal gametes, and if you use those abnormal gametes, then that could lead to abnormal individuals. So the previous slides were just showing you what you already knew about errors in meiosis. Sometimes you can have errors in mitosis. When you have errors in mitosis, they're not really going to be passed on to your kids for the most part because they happen once the individual's already produced. Errors in the gametes, well, you use the gametes to make the kids. But if errors happen in mitosis, that can lead to things like cancer, for example. So if you might have a healthy cell, perfectly healthy cell, it's going through healthy division for the most part of its life, and all of a sudden there is a mistake that happens, a non-disjunction error, and then that non-disjunction error is passed along to the other cells that are produced in the body, and that could lead to other serious disorders as well. So you've already seen this as well, but the idea is that you can do genetic tests to see if there have been any errors caused by meiosis and as a result ended up in the offspring. So for example, you can do a karyotype to see if you have an extra chromosome or a missing chromosome or missing segments, and that can lead you to see if your baby might have something like Down syndrome, for example. Now, another topic that I do want to speak about before we move on is the idea of non-disjunction leading to what's called polyploids. So in the previous presentation, we had learned the term aneuploid, which basically means having one extra chromosome or one missing chromosome. A polyploid, the difference in a polyploid and aneuploid is that the polyploid has multiples of a set. So for example, the polyploid, if I have a diploid and a mistake happens and it becomes a tetraploid, that becomes 4n. It gets two whole extra copies. The aneuploid only has an extra chromosome or a missing chromosome. It's not a whole extra set, or it's not missing a whole set. So polyploids have more than 2 sets of chromosomes. So you can see a triploid has 3 sets of chromosomes, 3 of each type. A tetraploid has 4 sets of chromosomes, 4 of each type. And that can often be caused by non-disjunction, so a mistake in meiosis that led to the polyploid being formed. So we can see, for example, wheat plants, they're hexaploids, bananas are triploids, and so I'll show you on the next slide an example of how this can happen, but I want you to get that it's not as common in animals as it is in plants. It is much more common in plants. Animals don't seem to survive this polyploid condition like plants can, but there are some cases. So here we have an example of a tetraploid mammal. This is a mammal here, so an animal, and it has an animal that is a mammal. You can see it has a larger tail. It also has larger sperm head than the diploid version, but the way it formed is that there were mistakes in meiosis. It made some gametes that were diploid, and when those gametes were used in some combination, it made a tetraploid offspring. And that tetraploid offspring was able to survive. So again, this is not an aneuploid, this is a polyploid individual, it's tetraploid. An aneuploid individual would have been something like 2n plus 1. It has 2 full sets plus an extra chromosome. The tetraploid has 4n. It has 4 full sets of chromosome. The extra set seems to be okay for the organism to have, or the extra sets seem to be okay for the organism to have and survive. It actually might be even better than having just that one extra chromosome. But the main idea is that a polyploid has whole extra sets as opposed to just one chromosome extra or missing. And again, that was caused by a mistake in meiosis during non-disjunction. We've also seen this in previous presentations where rather than have missing chromosomes, missing sets, or extra chromosomes, extra sets, you might have just damage to the chromosome structure itself. So you should go back and review this and understand how it can lead to developmental disorders and different phenotypes that we're not quite expecting. So you should be able to cite all of these different assumptions and then provide some examples of when these assumptions don't happen. So provide some exceptions to these assumptions. We've seen most of them so far. The only ones we haven't seen are F and I. I'm going to do a brief little overview of assumption F I'm going to leave I out of this for now. You can do a bit of research later on to figure out what it means, the gist of it. But for F, I do want you to have a general understanding of what that means as well. So one assumption is that genes assort independently. Remember, we saw that if you have, let's say, homologous chromosomes, so I'll draw homologous chromosomes here, and I'll draw some more here. These are two others. They're lined up along the metaphase plate. And let's say this homologous chromosome has big A on it, and this one has little a, and this one has big B on it, and its homolog has little b. Well, remember that we said that these chromosomes can assort independently, meaning that big A doesn't have to be with big B, and little a doesn't have to be with little b. Sometimes they could switch it up a bit. And so that's what we thought for the most case, that these genes could assort independently of each other. But what we now know today is that that's not always true. Sometimes you might have genes that are linked, meaning they're on the same chromosome. You can see that in this picture here. Big A is on the same chromosome as big B. So in that case, big A, for the most part, will have to end up with big B. They will not be able to assort independently. A is no longer independent of B because they're on the same chromosome and they're going to move together. So in cases like that, you might not get the same ratio that you would expect with independent assortment because big A does seem to have to end up with big B because they're on the same chromosome. So what we've noticed is that in some cases, yes, there are genes that are linked together and they do end up together more often than you would expect because they're on the same chromosome. You'll learn more about this when you get to university and you take biology there, but I want you to get that it's not one chromosome per gene. There's many genes on a chromosome, and sometimes those genes are linked, and as a result, they have to be inherited together. So you can see the example here. Here, the big G and the Y, they're on different chromosomes, so you can have a different arrangement about the metaphase plate. An independent assortment happens here just fine, and you get your variation of gametes. But if you look over here, we have big G and big Y on the same chromosome, so they have to be inherited together. Big G and big Y on the same chromosome, so they have to be inherited together. And so because they have to be inherited together, there's less variation in the gametes. Now, I do want you to know that this linkage is not perfect. Even though they're on the same chromosome, sometimes big G and little g might switch spots. When do you think that would happen? How could big G and little g switch spots? Because even though they are linked and we think that big G has to go with big Y, there are some cases where, yeah, those gametes of little g, big Y, and big G, little y can be formed. Think about that. What process could unlink the genes and create new combinations? If you guessed crossing over during meiosis 1, then that means you understand meiosis. Great. But again, for the most part in grade 11, we're just going to assume that the genes assort independently, and we're going to go along with that assumption. But just do get that you will see a lot more of this linked genes and unlinking when you get to the university level biology in university. So for homework, you should go through the worksheets and learning goals. Complete all the questions that are there using the PowerPoint in this video. You can also do some additional research. There's a great video in the learning goals and worksheet that you'll see along with some questions. This video is excellent as it tackles some of the assumptions that are made that simplify genetics a little bit and goes over the exceptions to those assumptions. It's about height and whether they're controlled or not by the environment, and you can take a look at the assumptions there. There's a few assumptions, so don't just assume it's just the environment affecting genes or something else in there that you got to look for too, and you can use the guiding questions to help you understand the video in more detail.